I'm supposed to everybody inform that it's being recorded now. Everybody will mute themselves unless they have a question, at which point they will unmute themselves. Here we go. Yes.
Concerning the quiz, can we skip back in case maybe
No,
after a question?
no. I will actually add that to the description. No, it's only one way. You cannot go back. You can only go forward. Okay?
Okay.
That was a very good question. Yes. All right. So today's lab, we're going to talk about introduction to laboratory growth media. They're also called culture media because that's what we use to culture or grow microorganisms inside a laboratory. And we'll also talk about common aseptic inoculation as well as transfer method. What this means is that how can we transfer microbe from one place to another aseptically using techniques that minimize or prevent any type of contamination at all. Next, we are going to talk about culture characteristics of bacteria when they grow on solid media, as well as when they grow in different type of liquid media. Culture characteristics mean when, we talked about colony morphology last time. So when you have a plate full of microbes growing on a culture plate, that's called a culture. It could be also be in a test tube, in a liquid or a solid medium. Nonetheless, it's called culture. Generally, I try to identify bacteria and fungi based on how they present themselves on the surface of a solid growth medium. So we will learn about what is the more terminology revolving around that. And then we are going to learn about some of the common aseptic transfer and inoculation methods. These are methods that ensure that when we are inoculating growth media with various microorganisms, we follow techniques that will avoid any kind of contamination, meaning you will prevent any unwanted microbe to be getting into that growth medium, and the only thing that should get into that is what you are intending to inoculate into that medium. Okay, so there's a lot of terminology that you notice I'm using. Inoculation, meaning to introduce microbes into a growth medium. Contamination means when unwanted microbes end up in a growth medium. And aseptic transfer techniques, they ensure that when you are inoculating growth media, you can avoid contamination. Okay, so this is the connection between all these concepts that we are going to discuss today. So let me move to the page where exercise number 2 is. Hopefully, most of you have had your printouts already. And if not, you should have a pencil and a pen handy and write down important points when I ask people to take notes. Okay. So week 2, lab activity 1, what does it mean to introduce, what are different type of growth media or culture media that we use, and what are some of the common aseptic inoculation and transfer methods? So as I said, understanding why we are doing any of these laboratory technique is probably the most critical part and in this setting. So the background information that I discuss here are critical for you to understand and learn, not only in terms of your quizzes, but also to set the grounds for any technique that are discussed in each these weekly lab activities. Unfortunately, we are not getting to do and much of the any of the hands-on work. So the best we can do is at least look at these. Talking about this, I will also take time to share some of the videos with you. We did not have time to watch the videos for Lab 1 last week because we kind of had a long meeting anyway. I hope that you guys checked out the folder with lab videos within the lab material folder, and had a chance to check out some of those YouTube videos. They are basically from internet that I have collected, and all that assortment is from YouTube. So there are videos related to today's topic where they actually show you how to do this in a lab. I have also assigned lab simulation in your McGraw-Hill website. I don't know how many of you had a chance to check that out, but the lab simulations are also available in there now. Okay, so what's a growth media?
Quick
Yes.
question. The lab simulations, do they have a due date?
No, I have set them up because I have not associated any grade with them. If you do actually take those, I may give some extra credit points at the end of semester if anybody needs it. But no, they are available throughout the semester because it is a lot of work that you have to do. So I kind of want to leave some of the things open that you can do in your time whenever you have time. So yeah, there is no deadline on them. They are available throughout the semester, and they are really good simulations. Once you try one, you'll know what I'm talking about. If you get stuck trying to figure out how to do it, excuse me, or anytime you need help with anything related to Connect, please call that 1-800 help desk number instead of sending me an email. They will help you out to resolve the issues. And now that I'm talking about it, I will also try to clarify that some students ended up getting registered for fall 2020 course instead of spring 2021. So they were not able to see the assignment and everything. I guided them to call help desk, and I hope that everybody has that issue resolved. If any of you are still logging into fall 2020 course, you just have to call McGraw-Hill 1-800 number that I provided you in one of the handout, and they will move you to the spring 2021 course. They are aware of the issues, and they said it could be because of the browser that students used or whatever the issue was, but you should immediately contact them so they can move you to the spring 2021 course. All right. So coming back to this, growth media are basically what we use. There's artificial preparations that we use in laboratories to grow microorganisms. We were unable to study or research or identify these microorganisms until the discovery of these growth media. So we are lucky enough to have a variety of growth media that are used in laboratory, in diagnostic laboratories, in research microbiology laboratories, where we grow microbes in these media, and that allows us to study them, that allows us to learn how to identify them. It allows us to conduct research in terms of how they change themselves, to do the drug testing and all sorts of things. In this topic, in this course, why is this relevant? Because in microbiology, infectious diseases are basic reason that you are in this course. So when we need to identify pathogens that are making any human being ill, when they go to see a doctor or emergency room or at a hospital, the body fluids are collected and they are inoculated onto various growth media. Once these specimens are sent to labs, and when they grow on these media, the microbiologist's job is to identify based on culture characteristics and based on metabolic characteristics, that what is the potential pathogen present in that patient's specimen. Okay, and that information is then shared with the physicians, which then can make a determination on what type of medication are appropriate to treat that patient's illness. Okay, so that is the main reason that you are being introduced to this concept in this course. Most of you are in nursing program and you probably won't be doing this, but you never know. Some of you may end up in a medical lab technician or technologist program, and then this will be your foundational course for that. So when we culture microbes, as you know, they're found everywhere. They have natural habitats, soil, water, air, on and inside living organisms, including plants and animals and all kinds of niches or niches where you can find microbes. But when we grow them in laboratory growth media, we call that growth preparation a culture. So if a culture, a microbial culture in a microbiology lab contains only one type of organism, it is called a pure culture. See this bold word over here, pure culture. However, most environmental specimen, most body fluid from patients, they do not generate pure cultures because there are multiple organisms present in these places. So whenever you prepare a culture from any of those, you end up with more than one organisms growing in it, and that type of culture is called a mixed culture. Okay, so what is the opposite of a pure culture? Mixed culture. And write down the definition. It should have been here somewhere, but make sure you write down the word mixed culture. So what is a mixed culture? A mixed culture contains more than one type of organisms growing in one growth medium. Okay, so that is the definition of mixed culture. Now these mixed culture could be just all bacteria, it could be bacteria and fungi and present all in one medium. Okay, so the media that we use, the word that I'm using, media, you see this word here, media, it's a plural of the word medium. So a growth medium is a culture that, like a nutritional rich environment that we use to grow microbes in. That medium could be in the form of solid or liquid medium. Okay, the medium, growth medium, could be solid or liquid. So this is what you have here, the word broth. A broth is a liquid growth medium. Okay, liquid growth medium is called a broth medium. And what makes a solid medium solid? That's the question. Why is a medium solid? So in microbiology cultures, we use this substance called agar. Agar is a complex carbohydrate which is kind of like, the starch. When you make your soup, you put corn, like corn starch to make your soup thick, right? So agar is a cousin of starch. So it's a complex carbohydrate, just like starch is. The difference, however, is that agar is so complex that almost all living organisms are unable to digest it. We do not have the metabolic enzyme to digest agar. You can digest starch, right? All that you eat, they have starch in it. All the rice you eat, they have starch in it. And we can digest them into simple carbohydrate, break it down into to the glucose subunits. But agar cannot be digested. Except for a very few microorganisms, nobody can digest agar. So agar is also used as a thickener in soups, in jams and jellies all over the world. And in fact, the introduction of agar to develop solid growth media was quite accidental. This microbiologist was trying to come up with a solid growth medium because he realized that the liquid media does not really tell us how many different type of microbes might be present in a broth, a test tube or flask or whatever you're growing it in. You cannot tell because they're all mixed with each other. So he wanted to see how you could separate various microbes present in a growth medium from each other. So he tried gelatin, didn't work because most microbes can digest gelatin. He tried cornstarch, didn't work because most microbes can digest. So initially the plate will be solid, but the following day when he will look at it, it would have turned into a liquid because the gelatin or the starch that he was using to solidify the medium will turn into liquid because they have been digested by microbes. So then this microbiologist's assistant, she handed him over this agar, and she said, you know what, I was given this to me by one of my Chinese friend, and it's used in jams and jellies as a thickener. Maybe you want to try this one. So they tried that to make a solid growth medium and inoculated with microbes, and to their surprise, it stayed solid forever. Forever mean for as long as they were observing it. And that was the introduction of agar to create solid growth medium. Okay. So if you ask me the difference between broth, a liquid growth medium, and an agar-based solid growth medium, what is the difference between them? Mostly the difference is just agar. Almost all growth media can be made as a liquid as well as a solid, as long as you add agar into it. Okay, so I hope that makes a distinction between solid and liquid. Next is introduction to some of the most commonly used ingredients in any of the culture media that we prepare. So familiar compounds such as sodium chloride, hydrochloric acid are used both in chemistry lab and in making media. Microorganisms are just like us. They need all the nutrients that we need, including macronutrients as well as micronutrients. They also need sodium chloride in small quantities, just like we do. Hydrochloric acid is used to adjust the pH, okay? Because just like microorganism, they also, the microorganism also need to have a certain pH to grow, and most of them like the same pH that we do, unless they are acidophilic or they like acidic environment, or they're basophilic, they like more basic environment. Most microorganisms fall into that near neutral pH, which works for our body. And if you remember, what is that pH that works for our body? It's 7.3, right? An equilibrium between 7.3 and 7.34. This is the range. You cannot deviate from this range. If you go down, you suffer from acidosis. If it goes up, you suffer from alkalosis. And both of those are pathological conditions. They can make us sick. So most microbes, they also like the same range, especially the ones that are pathogenic, right? They grow in our body because they have the same pH as they require. Most fungi, however, they like acidic pH. They will grow on this pH too, but they love acidic pH. So any pH below 6 is ideal for fungi to grow. Okay, so if you want to, let's say you have a sample from a patient and you only want to study if there are fungi present in it, you can choose to use a medium that has pH near 6 and only fungi will grow on that plate. No bacteria will grow because it's too acidic for them. Okay? So when you use such a medium that allows one type of organism to grow but weeds out the other one, it's called selective medium. That I'm gonna explain this a bit more later on. So a selective growth medium selects for one particular type of organism. Okay. So moving forward, what are some of these? We use beef extract, which is a protein source. We use peptone, it's also a protein source, and yeast extract. Because, as I said, microorganisms are just like us. Just like we need protein, carbohydrates, and all these other things, so do they. So when we are making these growth media, we want to make sure it has carbohydrates, it has all these, most of the protein sources. The thing with most microbes is they are, if you remember, you read it in chapter 1 and 4, they are referred to as chemoheterotroph. Meaning if you provide them with the necessary chemicals, they can pretty much chemoautotroph, sorry, not heterotroph, chemoautotroph, they can put together pretty much everything they need, especially amino acid, as long as they have all the basic ingredients. And then agar, water of course should be there. It's not, but you can add water. No growth media will be complete without any amount of water. Because even if a solid medium has water in it, because you dissolve everything in water, and then agar is used to make a solid. The thing about agar is once you prepare the medium and you let it cool off, it solidifies at 40 to 45 degrees Celsius, or room temperature. What I would like to explain here is that when you are preparing a growth medium, you guys see the drawings that I'm making, right? You're able to see my drawings, correct?
Yeah.
Okay, good.
Yeah.
So okay, good. That's it. I got it. So any growth medium that you're preparing, okay, you take a large flask. This is a glass flask. You add water to it based on how much medium you're making. Let's say you're making 1000 milliliters. So you'll add deionized distilled water. Then you will weigh everything. You will measure exact quantities according to the recipe, and you will add everything into this flask. Okay, your protein, your carbs, your minerals, your agar. Everything is added into this flask, and then the flask is kept on it's like a thermal plate that we used. It's like a thermal box, like a hot plate kind of. And then you will place this flask over this hot plate and increase the temperatures a little bit and place a magnet inside this flask. This plate also comes with as a magnetic stirrer, like, stir thing up. So once you turn it on using a knob on the side, the magnetic stirrer, it swirls around in your flask, and it will dissolve everything in the water. Okay, it will dissolve everything. Once everything is dissolved, then the next step is for you is to sterilize this growth medium. What is the next step? Sterilization. How would you sterilize this flask full of growth medium that you prepared? Whether it's solid or liquid, doesn't matter. How would you sterilize it? Any ideas?
Autoclave?
Autoclave.
Yes, very good, very good. So we will put this in autoclave for 45 minutes. Once it comes out of the autoclave, it's really hot. It's so hot, it's been at 121 degrees. You can't even touch it. So you put it on a countertop, you let it cool down, and when it comes to about 60 degrees Celsius, then it's still liquid. It has not solidified, but it's safe enough to handle it. Okay, so then what you do is you you take your empty sterile petri dishes or test tubes, whatever utensil you want to prepare your growth medium in, and then you pour that into your plates or your tubes. Tubes are generally filled up to here, and the plates are filled almost halfway through. And then you put the lid back on and let them cool off at room temperature. When it cools off to 40 degrees, it will solidify. Okay, that's how we prepare growth media. I would like to say, and then you can store them in refrigerator for for use. A lot of time they do just fine for a week in a refrigerator, but if you want to store them for a longer period of time, you may want to pack them in some plastic bags so they do not dry out. Otherwise, if you leave them just like that stacked up, the agar will dry because it's not airtight. The water evaporates through the lid. I would like to mention one thing about the tube media that I mentioned here. Once you pour this into your test tubes, a lot of time we use the test tube media as what we call slants. Slant. The word is S-L-A-N-T, slant. So in order to make this media to solidify in the form of a slant, we have to lay out our tubes at a 45-degree angle. So here's your countertop, and then you use, there's like a stand that we use in the labs where you could rest your test tubes at 45-degree angle. So in the agar, which was like a straight line like this, it turns into like a little V like this. And what you end up getting is this little, like a long layer of agar that gets solidified in your test tube in a slant. Okay, this merely increases the surface area for growth. When you use this to grow your cultures, you will have more surface area to grow your microbes on than if you just let it sit like straight up like this. It will solidify, of course, but then you will have just this much surface to grow microbe on. If you put it in a slant, it provides you with much more surface area. Okay, so that's a little bit of because when we are in a real lab, I show student all these things in thoroughly make media ourselves. It's already prepared for us, but I show them all the things that are in our labs that we use to make those media. Okay, so moving on, based on their consistency, the medium could be what? Solid. Remember, you have to add agar to make it solid. It could be liquid. What is the other word for liquid? Broth. And then there is something in between, as called semi-solid. So semi-solid is solid, but it's not as hard as the solid medium. So what is the difference? We use slightly less amount of agar when we are preparing semi-solid media. So now you can ask me, what is the purpose of semi-solid? Why do we have it? So semi-solid media are used to study, write this down, bacterial motility. Motility comes from the word motion, right? So a lot of bacteria, they are able to swim around, move in their growth medium because they have something called flagella. I'm sure you studied this in chapter 4, right? Flagella are means of locomotion for bacteria. They're different type of orientation that they could have for flagella, and we use different terms for those. But they are motile. They can move. So just by looking at bacteria, you cannot tell if they're motile or not. So we can use a motility medium like this one over here. This one is a motility medium. It used slightly less agar than this solid slant medium over here. You see the slant? Can everybody see the slant here? Right? Where I drew the blue line. That entire surface is good for you to grow your bacteria on. But this one over here is called agar deep. Agar deep. So when we are preparing a motility medium, which is a semi-solid medium, we use agar deep. Okay. If a bacteria is motile, it grows into, let's say, we inoculate it by doing something called stab. We stab into the medium. If the bacteria is motile, it moves around and it grows all over the tube. But if the bacteria is not motile, it will only grow where you stabbed it. It will only be restricted to that little area around the stab. But if it is motile, it will spread through the whole deep and grow everywhere. Okay, so solid media, semi-solid media are used to study bacterial motility. Did you write this down? Semi-solid media are good to study bacterial motility.
Yes.
Okay,
Esther?
good.
So
Yes.
to study the motility of bacteria, what is the point of knowing that?
Oh, very good question. Of course, this has significance in terms of not only diagnostic but also for research purposes and also for classification purposes. Okay, sometimes it's really important for us to know, like, you have two bacteria that are very similar to each other in all the other characteristics, yeah, but they could be different in terms of motility. One could be motile and the other could be non-motile, and that allows you, this feature, this test can allow you to distinguish between those two based on their motility. Got it?
Yes, thank you.
We're gonna use all of these things that I'm teaching you today later in our later exercises. Okay, we're gonna apply all these concepts. So then you will need to recall. This is really important lab that we are discussing. They're all important, but this lays out the foundation for a lot of tests and growth characteristics and things that we're gonna discuss later on. Good question. Good questions. Okay. So these three media that you see here, guys, if you have hard copy, you can label this. This first test tube is, of course, broth, right? You can see it looks liquid. It's very see-through. This one is agar, solid medium, agar slant. And the last one is what? Semi-solid. So these are three different types of media. Do you guys, nobody can see what I wrote?
Yes, we
The
can.
labeling on the picture? Okay. I can't. Oh, okay. Some people see it. Okay. Well, I don't know those who cannot see why you cannot see, because most people are seeing it. Well, you have to do refresh.
I've heard that it depends sometimes on the browser you use. You're not able to see what you're writing.
Browser, okay.
So maybe they.
Okay. And there may also be some time lag, right? Yeah. Anyway, first tube towards, if you're sitting facing your laptop, the first tube towards your left is the broth, the central one is land, and the whitish looking one is semi-solid. That helps if you cannot see it? Okay. Great. Moving on. Based on the ingredients, the media could be general, all-purpose. So what you're seeing.
The last one is what?
Pardon me?
The last one. I remember the first one is
Okay.
what?
The last one is the semi-solid. And remember, semi-solid is, as I mentioned earlier, is for motility. So the last one is semi-solid. This one here, C. Okay?
Okay, thank you.
Yeah, sure. So all of these that you see in this picture, they're all in this category. They're all general purpose media. This is probably the most common medium that we use. Did anybody raise a hand that I missed? No? Okay, so I see some people are coming in and out. I mean, there were 26 people at some point, and now I only see 24.
Maybe like
Okay.
me, some of them are refreshing their browser to see if we can see your writing. You see they're coming in.
Okay.
Yes, I went to refresh.
Sure. After this explanation, this topic, I'm going to stop to call roster, because remember that is really important for me to do that today for the VOE purposes. Okay. Moving on. So all growth. Oh, yeah. Sorry to hear that, but we have to live with that. What can we do? But I understand. If you were here and you got kicked out for some reason and you cannot get back in, send me an email so I know that you were there. I can also see that in my history, that who was logged in and how long they were here. I can also check that.
Can I make a little comment?
Mm-hmm.
So BCC is giving out like portable Wi-Fi's. I don't know if most of you have heard that, because I discussed that with my previous classes. Like most of the students doesn't know that. So I had my own last semester, and like I'm still using it this semester. It's really good, and since I got it, like I had no more issues with this Blackboard and network problem.
What did they give you? The Wi-Fi?
Yes.
Yeah, like the school is giving portable Wi-Fi's.
Yeah. Okay. Yeah.
Wi-Fi and a laptop.
So how do you access that? They send you a link?
Yeah, it's a link you have to just apply. They only need your employee ID, your business email, and then they just have to say you're taking classes.
Appreciate that. Okay. Can you post that in the chat so people who need it, they can copy it?
I can post it on our WhatsApp group chat, but I don't know how to post that on the
Okay.
If you share it with me, Samira, it's Ayana. If you share it with me on WhatsApp, I can share it on Blackboard.
Okay.
And if you could send it to me, like email it to me, that I can also post it in the Blackboard.
Okay.
So people have it. Okay.
Yeah.
Thank you. So should we continue?
Yes.
That was your comment? Okay, good. Thank you for bringing this up. All right.
Welcome.
So we're talking about all-purpose, different type of growth media based on their consistency. So the most common one is described here in this paragraph. I don't have to read all of this. I talked about these things already. But the most common one is the nutrient agar or nutrient broth. This was the one that you saw in the images on previous page, and this is also in the form of a petri dish. What is this plate called? Petri dish. It's after the name of the scientist who came up with it, actually. So P should be capital. Petri was the name of the scientist, but it's like a plate that comes with a lid on. Okay, so the image is actually the back side of it. But here is, let's say if this is the main dish, this is where you pour your agar. Let me use yellow color to fill it in. So you fill it in with this agar that is now solidified, and then I will use the red color to show you the lid. So then there is like a slightly larger, less deep lid part of the dish that goes over the blue part, and that is the lid of this dish. Okay, so once you pour the agar, you cover it with the lid and let it solidify. So that is your petri dish with the nutrient agar or nutrient growth agar. It's called nutrient agar. NA, nutrient agar. Nutrient agar plate. What's the name of this plate that you see in the picture? Nutrient agar plate. What you saw on the previous page was nutrient broth, nutrient agar slant, and nutrient agar deep. Those three tubes that you saw. So Iona has shared the link with guys. Anybody who is having issue with the link, copy that link and save it somewhere in your desktop. Okay. So beside the all-purpose general growth media, we also have special growth media. Special. The special growth media, there's so many types of them. It's impossible for me to share all of them, and also not the scope of this course. So I will just talk about a few of them. The special growth media are obviously, they are specific to promote growth of certain bacteria. So when you're using special growth media, they could be, okay. All right, guys. I know the link is really important, but I also want everybody to be paying attention to the lesson. Okay, I'll give you a moment to take these links down. Please copy the links. I'll stop because I want everybody to be paying attention to the lesson. Okay. So special media could be selective. Remember I mentioned earlier, if you only, okay, the break generally comes in the middle. We are only like 48 minutes into the class. Okay, so I will give you a break at about 2 or maybe 2:45, 1:45. Is that okay? So break will be 1:45. Remind me if I forget, please. Okay, special as well as differential. So these are really, really important terms for you to pay attention to, guys, as I walk you through the background. So a differential media, what does it mean? The definition is a differential medium have substances incorporated in it, enabling it to distinguish between bacteria, right? That's what the word differential means, to differentiate. For example, a medium called blood agar or trypticase soy agar, sheep blood, is a nutritive medium with differential properties in respect to hemolysis. Now, there's a lot of new words here for you to digest, okay? Underline hemolysis. What does that mean? So hemolysis, you studied your anatomy and physiology, so you are familiar with the word hemoglobin, right? And even with hemolysis, because you know the hemolytic anemia, I'm sure you heard the word hemolytic anemia. So the word hemolysis means breakdown of hemoglobin. What does hemolysis mean? Breakdown of hemoglobin. Yes, or erythrocytes. So now what does this mean for blood?
I'm sorry, Professor, what's that other word you just said, or what?
Breakdown?
You said hemoglobin, or?
I said hemoglobin, that's all I said.
Oh, I thought you said something else.
So she said, or hemolysis is the breakdown of hemoglobin.
Hemoglobin, that's what I said.
Okay. I thought you added another word.
No. So hemoglobin, there's no other word for hemoglobin. But you know, it's a protein, remember? Hemoglobin is a blood protein.
Yes.
Found, maybe this is the word that I mentioned, erythrocyte, the red blood cells.
That's what you said.
Yes. So it's everything I say is pretty much in this writing. I may not be like reading it exactly most of the time, but everything I say is in these pages. So if you skipped to note it, when you read it back, everything will be there. So anyways, erythrocytes are red blood cells, correct? So sheep blood have red blood cells in it. When bacteria grow on it, different bacteria have varying ability to do hemolysis of hemoglobin. Based on that, we learned that there are 3 categories to which bacteria can hemolyze the hemoglobin. It's called complete or beta hemolysis, impartial or partial or incomplete hemolysis called alpha hemolysis, and some bacteria do not cause hemolysis at all, and that is referred to as gamma or no hemolysis. So alpha, beta, gamma. Bacteria can break down the blood cell in this medium to 3 different levels based on their metabolic properties. So when a bacterium grows on this plate, looking at what kind of hemolysis this bacteria did, it can help us identify that bacterium, because we know exactly which bacteria cause beta hemolysis, which one cause alpha hemolysis, and which one cause no hemolysis. Got it? So what type of medium is blood agar? Differential. Why? Because you could study 3 different types of hemolysis on this plate based on the type of bacteria that is growing on it. Okay, and here is how it looks like. So this down here, let me switch to black color. This here is the actual color of the medium, red. You see that? Can everybody see my writing on the plate? So this is the red color. This part over here is where bacteria is growing. See, the red color is gone. That red color disappeared. Why? Because in this area, in the whole of this area, wherever bacteria are growing in this area, they have broken down hemoglobin. It's totally hemolyzed. Remember, the red color of the blood is because of hemoglobin. So if hemoglobin is broken down to amino acid level, the red color is gone. So we know which bacteria do this. This is beta hemolysis. Write it down. Label your plates if you have your hard copy. This is your beta hemolysis. And this is the original color of the plate. The reason this is still red is because there are no bacteria growing in this area. Now that was plate on left side, plate number 1. Plate number 2, look at the color. How would you describe that? One person, raise your hand. How would you describe this color on this plate number 2?
Like red?
Huh? Yes, Aona?
Red and green?
Well, red and green, yes. Everybody, I got it. So red color is, of course, the original color of the plate, right? Wherever you see red, that is because bacteria did not grow on those areas the way it was inoculated. Wherever bacteria are growing, you see these, all of these, this is the colonies, right? So wherever the colonies are, the color is green, or some people prefer to call it dirty green or brownish color. Okay, so this dirty green color is because of alpha hemolysis. This bacteria on plate number 2 could not break down hemoglobin completely because it's different from bacteria growing on plate 1. This one on plate 2 only hemolyzes hemoglobin to some degree, but not completely. So impartial or incomplete hemolysis, incomplete, also known as alpha. Alpha, this Greek symbol alpha, A-L-P-H-A. Alpha hemolysis. So plate 1 is beta, B-E-T-A, beta hemolysis. Plate 2 is alpha hemolysis. So microbiologists have a whole list of bacteria that cause beta hemolysis, and they have a whole list of bacteria that only cause alpha hemolysis. So this can help us differentiate bacteria based on hemolysis. Any question on this? Okay, nope. Moving on. Okay. So the next type of media is selective that I want you to know. Selective, as the word indicates, they're selective. Like, you know, you're selective in the food that you eat, correct? So just like that, bacteria are selective on type of media that they can grow on. And of course, that will depend on the ingredients present in a medium, correct? What type of carbs are present, what types of minerals are present, is there any specific amino acid present or not, what is the pH of the medium. There are so many features that we use to make different type of media selective. Okay, so selective media select what grows on it. So let's say, for example, if I only, okay, before I go into that, I want to make sure everybody understands the difference between gram-positive and gram-negative bacteria. You must have read that in your chapter 4. Okay. Anybody beside Iona want to raise hand and tell me what that means? What is gram-positive and gram-negative? Sorry.
Fine then.
Rosa? Yes, Rosa, go ahead.
So if I remember correctly, the difference is on the walls of the bacteria. So gram-positive peptidoglycan on their cell walls, but the gram-negative don't, if I remember correctly.
Say that again. Gram-positive have what? Peptidoglycan?
By accident. So they tint or color different colors when they're when you're being processing them. They stain different colors because the walls either lack or have I can't remember the name.
Peptidoglycan.
Yes.
Very good. Very good. You got it.
But you know which
so,
one which because of how they stain based on whether or not they have that on their wall.
Very good. Excellent. So bacteria, all known bacteria are distinguished into two, most of them, they fall into either gram-positive category or gram-negative category. And as Rosa mentioned correctly, the difference lies in the thickness of their cell wall. All bacteria, they have, their cell walls are made up of what is called peptidoglycan. Okay, peptido from peptide, which means proteins, correct? And then glycan, which means carb. So the cell wall are made up of this molecule that incorporate sugars as well as amino acid into it. So the whole world of the molecule is peptidoglycan. So gram-positive bacteria are the one that we recognize as gram-positive is because they have many layers of peptidoglycan. So their cell walls are very thick compared to this other group type of bacteria who have peptidoglycan, but they have very few layers of peptidoglycan. So their cell walls are thin. Okay, cell walls are very thin. So because of these cell wall differences, when we study these bacteria, we try to stain them in the laboratory using this technique called Graham's stain, given to us by this scientist called Christian Graham, and it's named after him. We call them gram-positive or gram-negative. Okay, so there is a medical significance of this concept, guys, and I want all of you to understand what that means. Gram-positive bacteria or gram-negative bacteria, we have human pathogen that fall into both of these categories. So when we are trying to diagnose a disease-causing bacteria in a patient, we need to know whether the patient has a gram-positive infection or a gram-negative infection. This will allow us to treat the patient better and correctly rather than giving, prescribing medication that won't work because we may be giving patient medication for gram-positive bacteria while they have a gram-negative infection and vice versa. So for that purpose, gram-positive, gram-negative diagnostic has a clear significance in medical field. So let's say you got a patient sample and you want to see if your patient has a gram-negative pathogen in it. You don't care about gram-positive because you know a lot of bacteria present in that patient body site are gram-positive. They're normal flora. You only want to see if it has some gram-negative bacteria. So instead of having to deal with so many different type of bacteria from that clinical specimen, you can use a selective medium, and that medium could be selective for gram-negative. So it will only allow gram-negatives to grow on that plate and will discourage all the gram-positive from showing up. So that will make your job easier. Okay, if there is a gram-negative present in the specimen, it will show up on the plate. If it's not, if the plate will not have any growth on it. So you'll know your patient specimen doesn't have any gram-negative in it. Did everybody follow that?
Can you repeat, Professor? I just want to be sure.
Sure. So the importance of selective media is in diagnostic, in clinical microbiology, as well as in research microbiology. If we are only interested in studying gram-negative bacteria, we can use a selective medium which allows only gram-negatives to grow and prevents gram-positive from growing on that medium. So that will make our job easier. If we use a selective media that selects for gram-negative bacteria only. Did you get it?
Yes, thank you so much.
Okay, sure.
Yes.
And then you can apply the same concept to vice versa. If you don't care about gram-negative and you only want to study a gram-positive present from a specimen, you can use selective media that are designed to grow gram-positive bacteria only. So that way you don't have to deal with the gram-negatives because you're not interested in that. Okay, so sometime you want to see them from one specimen, you want to see gram-positive and gram-negative, both of them. So you still could use selective media. So gram-negative will show up on gram-negative plate, gram-positive will show up on gram-positive plate, if a specimen has both of them present in it. So selective media makes a microbiologist's job really easy when it comes to separating things from specimens. Does that make sense?
Yes.
Yeah? Okay.
Yes.
Somebody wanted to say something. Go ahead.
Oh, I think we all said yes, that we understand.
Oh, okay, good. Great. So now here are some examples of some selective media, and it's very important that you guys know that for your quiz next week as well. Not only the definition of selective medium, but also some example. So here are some example of gram-negative selective medium. McConkey, EMB, also known as eosin, methylene blue, urea agar. These are some for gram-negative bacteria. There is a selective media for tuberculosis bacteria. It's called LJ. LJ stands for, it has a full word. I'll show you, it's in the picture next. Then we used this medium called mannitol salt agar, or MSA. This is selective for gram-positive staphylococci only. So this medium is extremely selective. So only bacteria that can grow on mannitol salt gram-positive, and not just all gram-positive, only staphylococci can grow on it. Understand? So there is a long list of selective media. We can't even go through the all list, but as long as you understand the concept, that's the main purpose here. But these are some media that we're going to be discussing in our upcoming labs. So down here you see LJ, or the selective medium that I mentioned for Mycobacterium tuberculosis we use. This is your LJ medium. And the full word for this is not written, so I will write it down. It's called Lowenstein. Write it down. Lowenstein or Stein.
It's reading on the second paragraph.
Somewhere, right? Okay, yeah. So it should be there. I just don't. Oh, here. Lowenstein-Jensen. It's in the beginning of paragraph. Okay, very good. Lowenstein-Jensen, for short, we call it LJ. This is uninoculated tube slant. As you can see, it's in the form of a slant. You see the slant? And the second tube is showing the colonies of a Mycobacterium tuberculosis growing on this medium. This medium is special because it has some complex lipids in it, waxy lipids. Because this particular bacterium need these lipids to be present in the medium to grow. So this is a unique medium, especially for this bacteria only. Okay, Mycobacterium, all species of this bacteria grow on this medium. I know that I've been showing using a lot of bacterial names today. I just want to make sure everybody understand how to, what is the scientific name. You learned about that binomial nomenclature in chapter 1, I guess. They explain that. Every scientific name of every living organism has two parts. So the first part is the genus name, right? Genus. Yeah. And the second part of the name is the species name or a specific name. Okay. So that's how we write. This is a proper way to write scientific name. It should always be italicized or underlined. The genus name always spelled out with capital. The first letter is always capital letter. The species should always be lowercase. Yes, Samira?
Did you say the end, the capital, like the beginning always has to be capital or the end?
The beginning, as you can see, Myco, M is capital.
Yeah.
Okay, so genus name, first letter is always capital, and the rest of the name has to be lowercase, including the species. Yeah.
So if you make it this way, like you don't need to underline it, but if you underline it, you don't have to.
Yes, yes. It should be underlined or italicized, never both.
Okay.
Somebody has was that Jones?
The mic.
Can you mute yourself? Because there's a lot of background noise. Miss Jones? Thank you. No, she did not. Okay. All right, so moving on. Okay, so you know about selective, you know about differential, yes? The next thing is that there are some growth media that could be both. Meaning one growth media could be selective as well as differential at the same time. Okay, so let's say MacConkey. Remember we talked about MacConkey? So MacConkey is one example. MacConkey is selective for what? What did I tell you? MacConkey is selective for gram negatives, correct? So when you have a different gram-negative bacteria growing on this medium, some of them make colored colonies, others make colorless colonies. So this medium is not only selective for gram-negative, it also allows us to differentiate between them based on if they produce color colonies or they produce colorless colonies. Okay, so just remember, MacConkey is selective as well as differential. Blood agar is only differential. So most selective media actually that we use, most selective media, they are also, not all of them, but most of them are also differential. Okay, so you can read the rest of the paragraph. That's what this is talking about. Let me go back to my next page. Here are pictures to describe what I just said. Here are pictures of various McConkey plate. You see the colonies, the little circles, and then here is like a thick layer of growth, right? This area, there is a lot of growth here, right? It's like layer of growth. But in this particular area, you could see individual separated little dots. Each one of them is the colony. But these colonies are colorless. They don't have any color. And look at this other plate, another McConkey plate with a different bacteria growing on it. And look at the color. How would you describe that? I don't know. Violet? Purple?
Purple.
Right?
Purple, or maybe.
Yeah. Yeah, purple.
Mm.
So same plate, McConkey, meaning same type of plate. It's not the same plate. They're two different plates, but they're both McConkey plate. This bacteria produced colorless colony. It's gram-negative but produced colorless colony. This is also gram-negative that produced purple colony. So that's what makes McConkey selective as well as differential. Down here is mannitol salt agar with plate divided into two, split into two by drawing a line at the bottom of the plate with a Sharpie. So half of the plate is used for Staphylococcus epidermidis. The other half, you're growing Staphylococcus aureus. Two different species of this genus Staphylococcus. Two different species, same genus. MSA, remember, it's selective for Staphylococcus only. It's a selective medium. But one species of Staph aureus, the plate is still pink. The other one plate turned yellow. Okay, so now we know, even if this was not labeled, and you see the bacteria is growing, the plate is still pink, you'll know the bacteria growing on MSA is Staph epidermidis. If your plate turned yellow, you know the bacteria growing on the plate is Staphylococcus epidermidis, even if it was unknown to you before. So just by growing these bacteria on these selective and differential media and how they grow, it helps us distinguish them from each other. Another selective and differential medium is this one. It's called urease. It is also selective for gram-negative, and it allows us to distinguish between bacteria that can break down urea, urease positive, and bacteria that do not produce urea, can break down urea, do not produce urease, sorry, can be distinguished based on color changes. So remember, that's why I told you colors are important, and I wanted you guys to have color copies. Okay, I hope everybody's okay. If not, you're gonna raise your hand, otherwise I just keep moving on. Based on gas or oxygen requirement, the media could be aerobic. Aerobic means has oxygen, provides oxygen. And the bacteria that grow in aerobic media, they are obligate aerobic.
It's 1:45.
Okay. All right, let me give you a break, and after the break, I'm gonna call roster. So please, everybody, stay here, okay? Don't go, because I'm gonna call roster after this. I'll see you in at 1:55. Okay, class, I'm back. So let me just call the attendance first. So I'm going to call according to the roster that I have by last name. So Ajay, Adjei, Bokeh. Did I say right? Is Ajay here? Ajay Bokeh. Okay, I see the person is there, but probably logged in and it's not. Cannot hear me? Ajay, are you there? If I don't hear from you, I'm gonna mark you absent. You can't just log in and just disappear. Ali Salihu? Is Ali Salihu?
Yeah, I'm here.
Okay, you're here. Good. Audi Samira? I know you're there. You are asking questions. Yes.
I'm here.
Yes. Ba Mariam?
Hello, I'm here. Mariam Ba.
Yes. Nina Boyki?
I'm here, Professor.
Awesome. Stanley, I know Stanley, you're there. You are asking questions.
Yeah, Professor.
Big Stanley, very good. How about Drame Fatu?
I'm here.
Okay. Were you here last week?
Yes.
Okay. Okay. Because remember, I logged out, my internet connection got disconnected, so the roster that I copied afterwards did not have your name. So if I don't have your attendance for last week, I'm gonna call everybody to a test. And do not lie, because I could check the log here. So,
I was here.
No, I know you were here. No, I know. I'm just saying in general, making a general announcement.
No,
Okay, Yahira Hernandez. I know you were there last week. Are you here today?
Hi, Professor. I'm here.
Okay,
yes,
great.
Can you hear me?
Yes,
Professor, I was here last week too.
Yes, I got you. Jones, Kamala Jones. Miss Jones, I know you were here because you asked a question. I know I could see you're logged in. Are you not paying attention? Kamala Jones? Miss Jones, are you there? Okay.
Professor. Kamala here. Sorry about that. I don't know what happened just now. I was trying to unmute myself to answer. However, it was still muted. So I just refresh and get back.
Okay.
Thank
were you here last week?
Yes, I was.
Got you. Because
Hi, Professor.
Yeah. Who is this?
This Boachi, I was having the same problem. That's why I left and come back.
Were you here last week?
Yes.
Okay, gotcha. John K. Are you there? I see that you logged in. Please unmute yourself and say hi. John? Tanya Livermore? Miss Tanya, are you here? Yes. It's okay. Were you here last week, Tanya? Okay. All right. Gotcha. forgive me if I don't say. Wyatt, are you there? Wyatt? I don't see you in the log. Okay, moving on. McLean? McLean Shadeen, you're here. Okay, were you here? You were here last week. I have you. Miranda? Is Miranda here?
Yes, I'm here.
Oh, Miranda, what is your last name?
I mean, that's my last name. Do you have Julissa Miranda?
Yes.
Miranda is my last name.
I'm sorry, what is it?
Miranda is my last name.
Okay, so what is first name is Julissa. Oh, okay. Because I think I had a different name in the login here. Are you using the same name here?
Yes, Julissa.
Signing up?
Okay.
Yes. Okay, gotcha. Good. And you were present last week. Nabu, Hannah? Is Hannah Nabu here?
Yeah, I'm here.
Okay, and you were here last week too?
Yes, please.
Okay, gotcha. Nadia Maya, I see you're logged in and you were here last week. Nadia, can you hear me?
Yes, I'm here.
Okay.
Thank you.
Okay. Got you.
Thank you. Thank you.
Sure. And next is Pinnock. Alyssa? Alyssa, are you here?
I'm here, Professor.
Okay. Were you here last week or you were the one who said that you didn't know if you have to be there?
Yes, I was here last week.
Okay, you were. Got you. Next one is Powell. Alia or Alia?
It's Alia. I'm here.
Yeah, okay. Alia is here. And Alia, you were here last Saturday as well?
Yes, I was.
Gotcha. Crystal Rodriguez.
I'm here.
Okay. And I have your last week's as well. And I know Iona, she's the ringleader, right, Iona?
Yes, I am.
Okay, Rosa, I know Rosa is here. She was asking question earlier. Sanchez, I got you, and I have you for last week. Sumaya, are you here?
Hello, Professor, I'm here.
Okay, and I have your last week's one. Next person is Leon Stedman.
I'm here, Professor.
Got you, and I have you for last week as well. Then I have Nadia, last name Oden. Nadia Oden. Nadia Oden, are you not here? Okay, I have her for
here.
Is that John?
Yes, please.
John, were you here last week?
Yes.
Okay. No, I'm just filling out my blanks because as I mentioned, explained earlier. And Aliyah, you were here last Saturday as well, correct? Aliyah?
Yes, I was.
Okay, got you. Andrea Washington. Andrea. I'm here and you were here last time as well. Got you. And next person is Tahani. Tahani, how do you say your last name, Tahani? Yafi, or how do you say that? Jafai? Is Miss Tahani here? Okay, she's absent today. She was here last week.
Professor?
Yeah?
I think she's having problem with the microphone, but she wrote in the chat.
She was? Okay. Thanks for letting me know.
Type in the chat.
Okay, good. Miss Nadia Uddin is not here. Let me just double check. No, she's not here today. And anybody in here that whose name I did not call? So Wyatt is absent. And other than that, and Uddin is absent. Other than that, everybody's here. So how many people do I have? 29 minus 3. I'm supposed to have 26 people. So 26 plus 1, it should be 27. And I'm missing 2. Correct. All right. I got you. I got you, Miss Tani. You're here. Very good. Very good. Thank you so much. I have my roster now. I know who is here. And so basically everybody is accounted for. Good. Thank you for your patience. Now let's continue with our lesson.
Professor?
Yeah.
Before we continue, I'm having a trouble accessing Connect. It's still showing me like the old.
Let's talk about at the end. Okay, I'll answer your question. Yes. Right. I already addressed that, but you probably were not here. But I will answer any Connect related question afterwards. Okay.
All right. Thank you.
Yes. Sure. So let's get back to types of growth media. So we talked about all-purpose, selective, differential, different examples of them. The next thing that I'm going to tell you is that, you know, we all need air, right? Fatu, can you please mute yourself? I just saw a question somebody had in the... Andrea has a question. I just want to make sure I'm understanding. Lab quizzes will be due every Saturday before midnight, not midnight, end of day, which is 11:59. So a minute before midnight. The lab quiz question will reflect what's on the printable lab papers. Yes, everything that I'm walking you through here, I'm explaining, all of those things will be the quiz be based upon. Is this correct? Yes. Connect question for multiple chapters are always due on the day of lecture exam, but of course they should be submitted before. When are the practice quizzes due? So practice quizzes if you're talking about the practice quizzes in the Blackboard, they have no due dates. They are all available throughout the semester. But of course, the idea is that you take practice quizzes. So when I give you actual lecture exam, the practice quizzes will help you prepare for your lecture exam. So you should be doing your practice quizzes for those chapters every week. And then you can always go back to them and do them again. For example, last week you should have done the practice quiz 1 on chapter 1 and 4. This week, you should do your practice quiz on chapter 6 as many times as you want. I hope that answers your question. Okay. Any other question about any of the housekeeping? Please write them down. I will answer them at the end.
Hello, how you doing?
I need to have continuity for this.
Hello.
Yes.
Hi, it's Miriam. I raised my hand. I wanted to ask you about the homework and the assignment. I get pretty confused. So,
okay, Miriam, all those questions will be at the end.
Okay.
Write it down, okay? I want to have continuity for this, and I have gone through all those things. There is a recording in the Collaborate for everything I was explained there. So if anything that you're unsure, of course, you can ask me again. But everything was explained in detail last week. So, but I will take your question after these explanations, all right? Because this kind of breaks the tempo for everybody. So any specific questions will be handled at the end. If you have questions related to lab that I'm explaining, that you can ask me now. Okay, moving on. We're talking about growth media now based on gas requirements. So what I was trying to explain was this concept of aerobic versus anaerobic. And being biology student, I assume that you understand the difference between them. These both terms are related to respiration, and in context of how do cells produce energy. Okay, the cells produce energy from glucose by breakdown of glucose. That's called respiration, cellular respiration, breakdown of glucose to produce energy. If they do that in the presence of oxygen, it's called aerobic respiration. And if it is done in the absence of oxygen, it's called anaerobic respiration. I hope that you know that from your previous bio courses. So microorganism, you know, human beings are aerobic. We need oxygen to live, and we cannot survive without oxygen for more than a few minutes. We'll die. That's because our metabolism will die. Our cells will not be able to produce energy. Our neurons, our brain, our heart will stop, and we will die. Bacteria, microorganisms, they are different. They have a wide range of gas requirement. In other words, they have different niches. Some bacteria are like us. They are aerobic. They need oxygen. They will die without oxygen. There are not too many of them. There are very few bacteria that are obligate aerobe like we are. We are obligate aerobe. We cannot live without oxygen. So there are very few bacteria that are obligate aerobes. Most bacteria, they are either facultative anaerobe, facultative. Write down this word, facultative anaerobe. Most bacteria fall into this category. So basically, facultative anaerobe, being facultative mean that you're very flexible. You have a wide range of conditions that you can survive in. That's what the facultative mean, having a very broad niche. So facultative anaerobe bacteria, they are basically aerobic, meaning they love oxygen. They do much better when they are growing in the presence of oxygen. However, if you take them out of oxygen environment, put them in an anaerobic environment, they will do just fine. They will not die. Okay, so that's, or sometimes for simplicity, we say facultative means false, or like, you know, like fake. So facultative anaerobe can live with or without oxygen. They do slightly better in the presence of oxygen. They will survive just fine when there is no oxygen. Why? Because then they will do anaerobic respiration. They could do fermentation, and they could produce their energy using those methods. So when you are growing microorganism in a laboratory environment, you want to make sure that you understand what type of bacteria are you dealing with. Is this aerobe? Is this anaerobe, or is facultative anaerobe? Having that knowledge will allow you to successfully culture your bacteria. For example, if you have obligate anaerobe, here, this one I'm circling around right now. If you have a bacteria that is obligate anaerobe means it must not have oxygen present. It can only grow when there is zero oxygen. Obligate anaerobe. If you're trying to grow that bacteria and you put it in regular environment, that bacteria is going to die. The oxygen is going to kill it. So if you have anaerobic bacteria, you must grow them in anaerobic environment or anaerobic media. If you have aerobic organism, you must grow them in aerobic environment. And if you have anaerobic, they'll grow anywhere. So having a knowledge of these properties about microbes and bacteria allows us to successfully culture them when we are trying to study them in a laboratory. I hear anybody has a question about this? Please go ahead. Okay, no question. So here is a picture of anaerobic media that I'm showing you here in on page 20. This test tube here. This is showing an anaerobic medium. It's called thioglycolate. Please, if you're not talking, mute yourself so that people can hear me better. Thioglycolate. What is thioglycolate? Thioglycolate is example of one anaerobic growth medium. Thioglycolate broth. Okay. So let's say you have an unknown bacteria. Yes. Yes.
Can you spell that? Because it's not. Did you type it on the screen? Because I can't see.
Oh, okay. Let me try text.
Okay.
Sorry. Can you see now the text box? It's written in the Word. It's right underneath here, or in the previous page, I guess. Glycolate. Okay.
No.
Did you get it?
No, there's no chat. I don't know.
Okay. Do you see the text that I typed?
No. Oh, got it. Okay.
I see the text.
Yeah, yeah.
It's not
Yes.
bigger. You gotta make a.
Okay. I could make a bigger font for the text.
You can also zoom in on your screen if you tap the view controls on the left side.
Yeah, I did. Here.
No, not
Can you see?
I'm talking about the class. If you can't see, you can always zoom it in.
Oh, the students. Okay. Yeah, don't try to do that because I think that affects my screen too. If you guys did that. But if it's working for you, that's fine. Can you see it now? Whoever asked that question. Can you see the word thioglycolate? It's written in the text too. Okay, when you read it, you'll find it. If you don't still see it. So anyway, if you have an unknown bacterium, okay, you have a totally unknown bacteria in terms of its gas requirement. You know, you got a patient sample, you don't know what type of bacteria is in it. You want to know what type of gas requirement the bacteria has. You can inoculate a thioglycolate broth test tube with that bacterium, and based on where the bacteria grows in this tube will allow you to determine what type of bacteria it is based on its gas requirement. From what I mean from that is, if that bacteria only grows on the tippy top, that will be an obligate aerobe. Because remember, this is an anaerobic medium. There is no oxygen here. Zero oxygen. There is some oxygen here on the top. So if your bacteria was aerobic, it will grow on the top layer in the form of it's a layer, literally. We call it pellicle, which means layer. So aerobic bacteria will grow in the form of a pellicle on top of the thioglycolate tube. If your bacteria is a obligate anaerobe, it will grow towards the lower half of the tube where there is no oxygen. Okay? Where there's zero oxygen, will grow here, obligate anaerobe. Then other things will be in between. And I'll show you that in a picture, but I want you to get this in your mind. Aerobe only grow on the top, obligate will grow all the way towards the bottom. And then facultative anaerobe will be growing throughout. It'll be growing throughout the tube, because remember, it can grow with or without oxygen. It will have slightly more density on the top, but it will just be growing fine throughout the tube. Okay? So there's another image that I'm going to take you, and then I'll come back to this page. And this is the image that I wanted to talk about, this one. You see this? So let's say that this is our thioglycolate tubes. Please mute yourself if you're not talking. Thioglycolate. Thioglycolate tube. Look, tube number 1, obviously. What type of bacteria is growing in tube number 1? It's only growing on the top. What type of bacteria is this? Huh?
Aerobic.
Aerobic.
Very good. True aerobe. True aerobe or obligate aerobe. It will only grow on the tippy top where there is plenty of oxygen. Number 2, what is number 2? It's only growing at the bottom where there is absolutely zero oxygen.
Anaerobic media?
No,
Obligate anaerobic.
Not just anaerobic. Obligate anaerobic. Meaning must grow without oxygen. Obligate anaerobe. Obligate anaerobe. Okay. So now between obligate aerobe, which is number 1, and obligate anaerobe, which is number 2, now these are other categories. So number 3, look at number 3. It's growing. Please mute yourself. Who is not muted? So number 3, if you see, there's slightly more growth on the top, but it's growing just fine throughout the tube. So what would number 3 be? Facultative anaerobe. Facultative anaerobe. Okay, see, they grow fine with or without oxygen. Although since they love oxygen, they will prefer to have it, but if it's not, they will be doing just fine. Number 4, there is less growth near the top, right? You see there's slightly less growth near the top, but just slightly below the surface, there's a lot of growth. And then there's nothing in the rest of the tube. So number 4 is a bacteria that doesn't like the amount of oxygen that is in the air. That is the maximum oxygen, right? The one in the air. But if you give it slightly less oxygen than the air, then it does better. So this type of bacteria is called microaerophilic. Micro, number 4 here. Micro means, aerophilic means it loves oxygen but loves less oxygen. Microaerophilic, loves slightly less oxygen. So that's number 4. And if you look at number 5, what do you notice about 5? Compare 5 with 3. What do you see? What is the difference between 5 and 3? Anybody? Alyssa?
At the top.
Say that again, Alyssa.
There's more clustered at the top, where 5 is kind of scattered throughout.
Throughout, same. Is same throughout, correct. Very good. That's a good observation. Now, the reason for that is because remember number 3, we said it prefers oxygen. So where there is more oxygen, it's reproducing much faster, but it's reproducing just fine in the rest of it too. But number 5 has no preference for oxygen. So the top, where there is more oxygen, has no meaning for this one. It's just growing same throughout without oxygen, just fine. Because you know why? The bacteria in the last test tube doesn't even know what to do with oxygen. It does not have the metabolic system to utilize oxygen. It does not do aerobic respiration at all. It uses other, remember how we talk about anaerobic respiration? So this bacteria can do anaerobic respiration, and it uses other inorganic ions as final electron acceptors, and we call this one as aerotolerant, meaning it tolerates the air, meaning oxygen doesn't kill it, tolerates the air, aerotolerant. So the aerotolerant, they grow just fine, because remember how aerobic respiration uses oxygen as the final electron acceptors, right? That's why we need oxygen, because that basically drives our respiratory pathway and our energy making systems like electron transport chain. But aerotolerant, they use nitrates or sulfates or other ions like that as final electron acceptors. So for them, oxygen doesn't exist. It doesn't matter for them. So that's the last category, aerotolerant. So which growth medium allows you to figure this out? Thioglycolate. Okay, now let me go back to and wrap up the types of media we were talking about. Semi-solid based on consistency and types. I explained that earlier. This is your agar deeps, uses only 0.5% agar. The regular agar medium, I think, uses 2%. This one only uses 0.5%. So it makes it really, really soft. So when you inoculate, remember I explained to you that the agar deeps or motility media, we use this inoculation equipment. It's called needle. Okay, so this is the handle. That's where you hold it from. And then there's like a metal needle, or sometimes we use plastic needles. I'll show you that our next activity is about that. I gotta speed up. So you pick up the bacteria at the tip of the needle, and then with steady hand, this is your agar, then you stab the bacteria into this agar almost like halfway through. You don't have to go all the way to the bottom. So this is how we inoculate growth medium. Now look at all these 3 tubes that you have here. Tube number 1, all the way to the left, bacteria was stabbed and it only grew where you stabbed it. You see the dark line? That means this bacteria only grew where you stabbed. It did not move into the tube because it has no flagella. It's non-motile. Tube 3, all the way to the right, bacteria spread throughout the tube. It did not stay only where you stabbed it, but it's growing everywhere. And it's because this bacteria has flagella and it's motile. The bacteria in the center is also motile. As you see, it spread away from where it was stabbed. The only thing is this bacteria like more oxygen. So this is only growing on the top part of the agar where it could get the maximum possible oxygen. But it's not growing that much towards the lower half of the tube because there was no oxygen for it. But it is motile. But Tube 1 and 2, they show you the extreme, non-motile and motile. Okay, then we use this media they call enriched media, and as the word indicates, they have some extra nutrients and some extra food that some bacteria must have to grow. So there are some bacteria that will only grow on blood agar or chocolate agar. They will not grow on your all-purpose media. So they need some ingredients that are present only in these media. So here's a picture of a blood agar plate and a chocolate agar plate. Chocolate agar plate does not have chocolate in it. It's just the color is chocolate. And it's basically a modified version of the blood agar. Blood agar have 5% sheep red blood cells. Okay? 5% sheep red blood cells in it. Then this last media I wanted you to know is called transportation media. I'm sure pretty much everybody is familiar with this. If you ever go to see a doctor, you're sick, you have strep throat, or if you go get your pap smear done, they use this type of media where the swab, this first part is the lid cover of the tube with the swab. That's where the specimen is collected, this part over here, and then it's placed into this transportation medium. The medium is on this part. It's inserted in there, and here's where the patient label goes, and then the specimen is sent to the diagnostic lab to be worked on to diagnose what's present in this patient specimen. The purpose of transportation media is that to keep the microorganism alive. If you did not use this, the microbe present on the patient swab, it will die by the time the specimen reaches to the lab. So transportation media are used to maintain the integrity of the specimen. They keep the microorganism collected from patient sample alive. They maintain the integrity. Not only that, they keep them alive, but they also come with some growth inhibitors, so they don't allow the bacteria to divide and multiply and increase their population in any way. So when the transportation media reaches to the lab, it has exactly the same number of microbes as they were collected from the patient's body. The reason we do that is, let's say you collected a patient sample, there are bacteria that are normal flora from that body site, and then there are few that are pathogenic. So what will happen is, if you not prevent them from multiplying, the normal one, they will continue to multiply, and they may kill the few pathogenic cells that you collected from your patient sample. And the test will come out negative. So in order to avoid that, microbiologists learned that it's best that we collect the specimen in this transport media. The list of transport media is long. There are specific ones for specific purposes. There are different ones used for anaerobic bacterial collection. Remember, anaerobe will die if you expose them to oxygen within half. So transport media vary based on the purpose, and I'm giving you some example here. There's a Stewart medium, and then there's a Carey-Blair medium, which has a buffered glycerol saline. There's a SEP, and it's only used to collect Salmonella and Shigella specimen. There is a different one for gonorrhea specimen. So the list of transportation media is pretty long, but you get the idea, right? Okay. Next activity is to familiarize on how to distinguish colonies from each other. Because there's something called microbial colony. I gave you a definition last time, right? It's an aggregate of microbial organism on the surface of a solid growth medium. Bacteria make colonies on surface of solid media. Fungi make colonies on the surface of growth media. You have to remember that we cannot grow viruses in these media that we are talking about. They are not to grow viruses, because viruses only grow inside living cells. So the things that we are talking about, they are for bacteria and or fungi. So when you are looking at the colony, you're basically looking at either bacterial colonies or fungal colonies. So what are some of these criteria that we use? Okay, we call them colony characteristics. Colony morphology. Morph means shape, right? Morph. Morphology means study of shape. So colony morphology, or we also call it colony characteristics. So what are they? Form. What's the other word for form? What would be the other word for form? Shape. Form means what is the basic shape of your colony. The colony could be circular, they could be irregular, they could have thread-like edges, so we call them filamentous. What is elevation? Is the colony flat? Is lying flat with the surface of your agar, or is it raised? So if this is the surface of your agar, if the colony is lying flat, it will be like sticking to the surface of your agar. But if a colony is like this, it's called raised. There are different variations of raised. I'll show you that in a second. Margin mean what type of edges the colony have. Are they like clear lines, or they are zigzaggy, they are wavy, or they are like thread-like, filamentous. So that's the margin. Then you look at the surface. Is the surface smooth, glistening meaning shiny? Is it rough, dull? Or sometimes there are bacteria whose colony surface is very wrinkled. These characteristics can help us identify which bacteria this might be. The other thing we look for is the optical properties or opacity of the colony, which means, is the colony clear, see-through? Is it opaque? Or translucent means clear or see-through. Is it iridescent, meaning does it have like very special shiny things to it that are reflecting the light? Does it have any color, pigmentation? That's another thing we look for. Okay, so form, elevation, margin, surface, opacity, pigmentation. What are these called? Colony, morphology, or colony characteristics. And this is what we mean by that. Look at different shapes. Edges of the colony. To look at the margin, you look at the edges. See, entire margin means that it's a very clean line. It's like a perfect circle, like you took a compass and drew a circle with it. Undulate means there are wavy. Lobate is a type of undulate, but the waves are very deep. Curly, even curls. Rhoizoidal, root-like, or thread-like edges, meaning filamentous. Elevation, flat. Raised. If the raise is like raised, but the top is like a level field, then it's called raised. But if it goes up and down like a mountain, it's called convex. If the convex has a very like a steep curve to it, it's called pulvinate. And if it has like a camel-like hump in the center, it's called umbonate. But all of these are types of raised colonies, different shapes. Different bacteria present themselves in only one of these forms. One particular type of bacteria will always have the same morphology, will not have different shapes. Size, punctiform mean like a needle point. Like you took the back of the needle and you just like, touch the surface of agar. So they call needle point or punctiform. Small, moderate, large. Texture could be smooth or rough, dry or moist, mucus-like or wrinkled. Appearance could be glistening or dull. Pigmentation will depend on color, white, cream, tan, purple, red, yellow, whatever would be the color. And optical mean, is it opaque or is it see-through? Okay, so what are all of these? Colony morphologies. Before I go to the next thing, guys, I want you to go back on in your lab manual to this page 14 and study these colonies. Look at that. Let's describe one of these colonies here. Sorry. Which one should I pick? Okay, let me pick this colony. You see this pinkish colony here, or orange? Does everybody see that? Where my hand is, the cursor is? Yes, okay, that's enough. Not everybody has to say yes, I got you. So how describe this colony? Mariam, do you want to describe it?
Like describe how it looks?
Yeah, based on the terms that we just went through. Let's start with shape. What do you think is the shape or form? Circular, right? Isn't it a circle? Yes, circular. What about the size? Is it punctiform, small, medium, or large?
Medium.
Medium. Very good. And in terms of the elevation, does it look flat or does it look raised?
Look raised.
Yes, very good. It's raised. And can you tell what type of raised? It's like this. Convex. Convex. Does it have glistening look or dull look?
Don't look.
No, it's glistening.
Oh, okay.
Glistening.
It's glistening. In terms of opacity, is it opaque or see-through?
Opaque.
Very good. So now you could put all of this together. Oh, the margin. Does it look like a clean line or does it have edges like lobes or wrinkles or those things?
Clean line.
Clean line. Very good. So everybody, we are going to describe this colony now. Let's see. Circular, entire margins, raised, medium, paste-like or glistening, opaque. Very good. And last thing is the color, right?
Yeah.
How would you say it? This color is called salmon pink in microbiology. We call this salmon pink colony.
Okay.
So that's how you describe a colony. Got it? Now let's pick another one. Let's pick this white rough one here.
Pick a bigger one so everyone can see.
Huh? What is it?
I said pick a bigger one so everyone can see.
Yeah. Well,
you can enlarge your screen. On the left-hand side, you see where you have the plus sign and then the negative sign.
It's the maximum.
And then the arrows, and it only does it to your screen. It won't affect no one else's screen.
No,
No, not you, Professor. If you do it, we see it. But if the students do it, you don't see it and no one else sees it. You can press the plus sign on your screen and it'll make it larger for you.
Okay, did you guys all hear that? Thank you, Evna. So here is a bigger one now. But if you notice, there are many tiny ones buried underneath this bigger one, but ignore those. I'm only talking about the large one. So this one, what is the shape? It's circular, definitely. But is it? Does it have entire margins? Not really. Irregular. How about the size?
Small.
No.
You talking about the first one, the first circle?
Oh, the one where my hand is right now. Do you see my hand?
This one.
Okay,
no.
yeah. It's towards the right. The big white one towards the right.
The bottom right. Okay.
Yeah. See, somebody drew a line and it showed up here.
Yeah, I saw it.
Okay. So it's large. It's large, okay?
Yes.
So circular, irregular, and it's kind of flat. It's not raised. Flat, white, opaque.
It's opaque? Looks
yes,
transparent.
It's transparent.
No, no.
We can see the stuff under it.
You can see the little.
No. You can see underneath because of the consistency and color of those colonies. But can you see the counter through it? Can you see the black counter through the colony?
No.
Then it's opaque.
Okay.
Okay. See-through mean that you could see all the way towards the bottom of the plate. That would be translucent. All of these colonies are opaque. All of them on this plate, they're all opaque. Okay. So that's how we describe colony, guys. Now look at the mold colony, any mold colony that are here. So I want you to label this picture with me, guys. Okay, first look at this. This is a labeled picture. So the yellow one, and this is actually an error. Just ignore this one. But the other two are yellow colonies here, right? You see the color is yellow. They belong to this bacteria called Micrococcus. This is the salmon pink one that we described earlier. This bacteria is called Serratia. This is the white one, kind of like the white one we just went through. See how the edges are undulate? And this bacterium is called Bacillus. And then this cream colored one over here, which is kind of medium, small to medium, it belongs to this bacteria called Escherichia coli. So using these labels, please mute yourself. So using these label picture, guys, okay, Nina, please mute yourself. I could see from here who is not muted. Okay. Just like you, okay, so here. Hello? All right, coming back to the picture. I want you guys to label this unlabeled plate using what I told you in the picture above it. What is this white bacteria? What is its name? The white colony that we just described. This one would be Bacillus. Can you identify many other Bacillus colonies on this plate? Like this is Bacillus, this one is Bacillus, this one is Bacillus, many Bacillus colonies. Because this is one of the most common bacteria found in our environment. Can anybody tell Nina to mute herself?
Professor, I did. I don't know why. It's muted, but I don't know why you guys can still hear me.
No, okay, let me mute you. Now you're muted. Okay.
Professor, you can just mute everyone just in case.
Yeah. I should do that. Okay. So, where was I? Okay. So, what is this yellow one based on the plate above that? The yellow one? This yellow one. Very good. Very good. So, label this Micrococcus. And what is this pink one, salmon pink one down here?
Serratia
or Serratia. You could pronounce it either way. Okay, and what about this velvety green one down here? Look at this one. Remember, these are different molds. So this velvety green one, this belongs to a mold called Penicillium. Penicillium. Okay, so that's this one, Penicillium. This is Penicillium. This is Penicillium. This is Penicillium. This is Penicillium. This one is Penicillium. This is a different mold. This is another mold. Different. This is also Penicillium. Okay, so how do you distinguish Penicillium colony? Based on the color, size, texture, just like you do for bacteria. Is everybody good with colony morphology? Can we move on to the new topic?
Yes.
Go ahead.
Yeah, how do you tell if it's raised or not?
Okay, very good question. Unfortunately, I cannot show you this here, but there are videos, and I'm not sure if we're gonna be able to get to the video. So you're gonna watch the videos in your own time. Everybody promise me. I'm gonna check who accessed the video and who didn't. must watch the videos, and they demonstrate all these things, including how to study this, the elevation. So
You mean
unfortunately,
the lecture videos?
What is it?
Are you talking about the lecture videos?
No. In the lab material folder, there are YouTube videos about each lab.
Oh, we have videos for labs too?
Yes.
Oh,
They're
I only saw
YouTube
the latest
videos.
one.
Yeah, you're gonna watch them. I will show you where they are before you guys leave me today, okay? So everybody should watch, and they demonstrate how to study elevation. That was a very good question, Sumaya. So what you do is you hold the plate that you are studying at the eye level. You hold the plate, you hold the plate at your eye level, and then you look at the surface of the colony, and that tells you, you can see very clearly if the colony is flat or raised. Fortunately for us, most bacterial colonies are raised. What was that? Are people having issues? Okay, alrighty. So now going back to the...
Will we have to identify the colonies like on an exam or a quiz, because it's harder to see
No,
like here.
No, but I will ask you a question about the image that I just showed you about colony morphology.
Okay.
But there will be one or two questions about that.
Okay.
But based on how I described to you, I think once you actually have a plate in your hand, you should be very easily. But there will be question from this image though. I may give you one of these and ask you to identify what type of elevation it is or what type of margin you see in the picture. Is that fair?
Yes, if we're able to see it. If we can see, like, because it's hard to see if it's raised or not just looking at it from the top.
No, I'm not going to ask you that. I said I'm going to use this image, this type of image. Like, maybe image of one of these margin or one of these elevation from here, not the plates, no.
Okay, thank you.
Okay. Okay, so continuing with our lesson, we already went through this page, so I'm not gonna spend time on this again. The thioglycolate one. I think my internet froze, so give it a second. What happened? Why did you freeze on me? Can you guys hear me?
100%.
Yes.
Yes.
Okay, but for some reason my slide that I'm sharing with you, everything else is frozen. So it just gave me a message that it's trying to link again. It's taking a long while. And I don't want to, I called the attendance already, so that's not an issue. Okay, it's doing something. My internet connection is fine. Blackboard is not responding. So if you can still hear me, I'm gonna log out and come back in. Stay in there.
Okay, Professor.
Stay in there, don't go anywhere. I'm back. You guys hear me?
Yeah.
Yes.
Okay. Okay. All right. Great. 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 So the last but not least is the introduction to what we mean by aseptic techniques. Okay. So aseptic transfer techniques mean, and again, there is videos on how to do this as well. So a person is actually sitting down and going to show you how all of these steps that I'm going to introduce you to in this activity. So basically what you do is you sit down with your microbial culture. We give student test tubes that have cultures growing in it, bacteria growing in it, and then we give them fresh growth media. So they take bacteria from the media that we give them, culture we give them, and inoculate their plates and their test tube with solid media and test tube with broth. So they get to practice all three types in this exercise when we are in person. And so I introduced you to growth media already. In this exercise, I'm going to introduce you to the inoculation tools. Inoculation tools that a microbiologist uses are just these two here. It's called loop. One is called loop, and if you could see, you could maximize on your end, but you could also see it here. A loop is just like a metal, it has a metal holder, and then a needle which has like a little circle at the end. Can you see this little O-ring here? So that's your loop, and needle is just a straight needle. The needle is also called, we're also generally microbiologists use the word pick for them, because you use this to pick microbe from one place and put them in the other. Needles are, you can use either one of them, it doesn't matter. The loop has a little bit of advantage that it picks up slightly more amount because it has like that little circle in it. So metal, loop and needle, this is what we use in our lab. These are reusable. You are supposed to sterilize them before and after every use, either by burning them in the Bunsen burner. That's what you'll see in the YouTube video. You hold it in the red part of the flame to sterilize, or then there's something called, it's very small, it's like a very small furnace. It's called incinerator. Incinerator. From incineration, to burn. So we use either incinerator or the Bunsen burner flame to sterilize the metal loop and needle every time we use it, before and after. Most laboratories use these disposable plastic loops in the second picture on this side here. As you can see, two yellow needles, and then there are various loops. And if you read at the top of each loop, it has a number written. It says 1 microliter, 5 microliter, or 10 microliter. So these loops that have this number written on them, they call calibrated. What is the word? Calibrated. So calibrated loop means that when you use this loop to pick up a liquid growth medium, it picks up 1 microliter of the fluid in this loop. And if you use 5 microliter, this one, it picks up 5 microliter of the fluid, and the 10 microliter picks up 10 microliter of the fluid if you're working with a liquid growth medium. That's what they are used. Sometimes we use them, as I will explain when we go into other exercises. No need to spell everything out today. Okay, so loop or needle, they are used for sterilization. You have to sterilize them. There's a whole bunch of terminology that revolves around this topic. What we mean by aseptic. Aseptic refers to sterile transfer method, meaning you do everything that you can to avoid contamination. I already explained to you contamination mean introduction of unwanted microbe into your growth medium. Inoculation means to introduce microbe into a growth medium. Your growth medium could be solid or liquid. It could be a plate, a petri dish, or it could be a nutrient broth, or it could be a nutrient agar slant. The process then talks you through it, so you can read it, but basically it's telling you step by step of how you inoculate. So the steps are pretty long here. I don't particularly ask you in the quiz about the steps on how to do it, but I want you to give this at least one reading. Make sure you go through all the definitions that are here. Single clone versus pure culture versus a mixed culture. And last but not least is this concept that I want you to talk about, is about streak plate. So when you are inoculating your growth plates, there is this method that we use in microbiology. It's called streaking. Everybody understand the word streak. It's like a regular English word, streak. So you have your plate. Let's say this is your tube that has E. coli growing on it, and you want to inoculate now this petri dish with E. coli. So what you'll do is you'll take either a loop or a needle, pick up small amount of E. coli from here, and transfer it to your plate by going over the surface in this first part of the plate. That's called streak. Then you will, using a Bunsen burner or your incinerator, you will heat up the tip of your loop or needle that you're using to get rid of excessive inoculum, and then with that loop or needle go back into this first streak that you made a couple of times, and then streak into this next half of the plate. See how the streaks are not touching each other. Then with the same loop or needle, no need to heat this time, you go back into the second streak couple of times and then finish streaking on this part of the plate. So this is called streak plate method, which is the most common method used in microbiology when we inoculate plates. It allows us to separate bacteria from each other when you put them on the surface of the plate. Instead of them clumped on each other and growing on top of each other, and then you wouldn't be able to tell how many bacteria are there, what type of colony morphology they have. Streak plate allows them to spread them out and kind of try to separate them from each other. If you are working off of a mixed sample, it is must that you do a streak plate method. Sometimes you have to repeat the process twice. If the medium, the culture that you use, the sample that you use was very, very condensed, there was too many microbial cells in the liquid, it may not be possible for you to separate them out in the first plate that you do. So then you may have to kind of pick them, a few of them here and there, and put them on separate plates and try to separate them in a second plate. It's called subculturing. So on the net, it talks you through the steps that I just explained, that you can do that. When you are new to this, what we ask students to do is, behind at the back of their plate, we give, with the help of a crayon or a drawing pencil or a Sharpie, we write the word T at the bottom of your petri dish. This allows you to separate your plate into 3 separate areas. So it allows you to make sure that your streaks are kind of organized. So first streak goes into this first area, you just go over the surface. Second streak goes in this area, and third streak goes in this area. So drawing a tree kind of help you when you are new to master the technique. But once you've done it a few times, you don't need to draw the T. You could just go and have a nice streak here. Once you grow the bacteria from after you inoculated, you look at it next time. Look at this plate that you see. It's telling you there are more than one bacteria definitely in this plate. You probably notice that you always go clockwise when you are streaking. You never go anti-clockwise. You're always supposed to go clockwise. This first streak that I showed you is called a 3-sector or 3-sector streak. Then there is a 4-sector streak shown on the next page here. So when you're new, you try 3-sector, but then you can graduate on to, when you develop that kind of motor skill, you could also do 4-quadrant streaks. So the different color on this plate don't mean that there's a different bacteria being put on here. It's just showing you that this is how your streak 1 goes. Streak 2, you notice how you just go a couple of times into the previous one to pick up the inoculum, and then you spread it onto the 2nd quadrant of your plate. 3rd streak is shown in purple here. You go back to the 2nd streak only a couple of times and then continue to spread it into this 3rd area. And then the 4th one, you only go into the once, and then you spread it out onto the rest of the plate. So if you look at the result of a streak made using this technique, look at the next picture next to it, the white bacteria growing on it. In first area, see colonies are so superimposed, you can't tell them apart from each other. 2nd streak is also like that because there's a lot of inoculum, a lot of cells that you're putting in those areas. So they're too much growth. Each colony get merged with the next one. You can't have. 3rd one, there's slight separation. But the 4th one, you could see beautiful single stand colonies of this bacteria, which then you can use to start pure cultures, right? Because you could see that there's just one type of colony, only one microbe growing in this medium. And you can pick one individual colony, inoculate that into a fresh growth plate or tube, and you will have a pure culture. Got it? Look at this plate down here. This is how our plates look like, because what we do, we give our student a mixture of E. coli and Micrococcus, and sometimes we actually throw Serratia in there too. So we give you a mixture of 3 bacteria, and then you practice this 3 or 4-sector streak technique. And by looking at this, I mean, the 1st and 2nd area, they're no good because they're all growing on top of each other. You could tell clearly that there are 2 different colonies in here, right? The white one and the yellow ones. But looking at this 3rd area, now you have separated colonies of E. coli and separated colonies of Micrococcus. And so you could pick each one of them and start a pure culture. So in a nutshell, what is the advantage of streak plate? It allows you to separate microbe from a mixture so that then you can start pure cultures once you get separated colonies. It is also the most commonly used method to inoculate plates worldwide. Okay, guys? So you're going to answer all of these questions that are at the end of each one of these activities to prepare for your quiz. You're also going to watch the videos. Let me show you where the videos are. And then you can watch videos on your own, and then I will love to answer the question that you guys may have. Let me just show you the video folder first. So I'm gonna stop sharing file. I'm gonna say share screen. And I think I have this Blackboard window open. It's getting there. Okay. Do you guys see the No, I think you keep sharing this screen. Stop sharing. Share screen application window. I think it's this one. Share. Okay. So you see this is the Blackboard site, your Blackboard site. Can I make it bigger? So you see this folder says lab materials. In here, this is the folder called lab videos. Just one person tell me, do you see it? Do you guys see my screen?
Yes, Professor.
Yeah.
Okay. Okay. So this is the lab videos folder. See, there are lab safety video. This is for last week's lab. And then here is colony morphology growth pattern videos. You're gonna watch that. Aseptic transfer colony morphology videos. And more aseptic transfer videos. These are all really, really useful videos. They're not long, they're a few minutes each. So please watch these, okay? Because I love to take time to explain things. So unfortunately, I may not always have time to show you videos, but they are all here for next week's lab. Here's a gram staining video, acid fast videos. So there are videos for pretty much all the labs that I'm gonna do, okay? Make sure to watch these. I'm gonna stop sharing, and now you could ask me any questions that you may have.
Okay.
I know.
Hello. So, Professor, this is Miriam speaking. How you doing? So
I'm good. How are you?
I wanted to ask you the work that you just told us to do is where can we send it to? So you said we have to do the questions.
Which questions? The practice questions?
No, the question that you just said on the lab menu.
No.
Oh no, you're just gonna study them to prepare for your quiz. You don't have to submit to me.
Okay,
You're gonna
okay, yeah. I know I'm gonna practice on it. So for the homework and the assignment, so on Connect, it's when you have to do the lecture work. Everything is due on February 20th.
No, everything for lecture exam 1. Lecture exam 1 includes chapter 1, 4, 6, 7, and 8. Or I think probably 9, I'm not sure. Those are due on February 20th. Before you take your lecture exam.
You said February 20, what is it?
20th. That is the day you have your lecture exam 1 on Blackboard.
Okay, but I'm saying for the homework, what is on all the homework is on Connect?
There is lecture homework on Connect, yes. And there are 2 types of homeworks on Connect, okay?
Which is
lecture, textbook assignment. The Learn Smart, you read the chapter and you take all the questions that come with that chapter. Once you complete all the questions, you get assigned 100 points in Connect for that chapter.
Okay, and everything is due by February 20th, right?
What do you mean everything? Everything for exam number 1.
Because on the Connect, it's saying that all the assignment is due by February 20th.
Yes, for lecture exam 1, yes.
Oh, okay. So the lab is there too?
Lab, for the Connect, there is a tab, it says lab simulations. So those are optional. They're not compulsory. If you do that, I will give you some bonus points at the end.
So lab work and Connect is not required?
No, it's optional.
Okay, but it's good to do the practice quiz so you won't get points for the final. Just the practice quiz that's on Blackboard, correct?
Yes.
And the Connect is just for lecture work, basically.
Yes, yes.
Thank you.
Got it. Sure. Any other question? I know, Fatu, you had a question earlier. Do you want to ask?
No, I was, my question was about Connect.
Did I answer that already?
No, you said, you told me to wait till like when the class is.
No, now I'm asking you to ask now.
Oh,
what was
yeah.
the question?
Yeah, I can't access our class. I think the class that I'm in is the previous one, the 2021.
Yes. So here's what you're going to do. You know that document posted on Blackboard that says syllabus language?
Mm-hmm.
It has a 1-800 number written there for you to call.
Okay. All right.
And tell them that somehow you are ending up in fall 2020 course for Shazia Khan. Tell them my name. They have my information and they know that I've reported it to them already. Tell them, can you please transfer me to spring 2021 course?
Okay.
And they should be able to do that for you. Okay?
All right. Okay.
Yeah.
Thank you.
No problem.
All right.
Nadine, you have a question?
Yeah, hi, sorry. It's just that the thing with the lab simulation, it doesn't, when I click on it, it just goes to an error page, just so you know.
Okay, I'll double check.
Yeah, I've tried a couple times because I wanted to go in and see what it was about.
Oh, okay.
Yeah, but it just
Yeah,
goes
they're kind of weird. Yeah, they're kind of weird. That's why I didn't make it mandatory.
Mandatory. Okay, no problem.
Yeah, it's not mandatory. So don't worry if it doesn't work sometime.
Okay, thank you.
Yeah. Sure. Okay. All right, I get it. I'll look at it and make sure that they activate them. It's like a long process to be deployed. So maybe I have to go through the deployment again because I thought assigning will make them active, but obviously that didn't make them active. So I will have to deploy them again. I'll work on that. Okay.
Thank you so much. And Professor, in the week 2, the lecture practice thing, it doesn't say that it's like multiple attempts.
Okay.
It just do the one attempt. So I just want.
All right. Let me do that. Let me do that right now.
Thank you so much.
Before I leave. Yeah, no, I'm gonna do it right now.
Can you see the grade? Can you see the grade of the practice exam?
Say that again, please.
For the practice exam on the on Blackboard, I did it only one time and I have 100. I was asking, can you see the grade?
Yes, it shows me the grade. But remember, okay, there's something I wanted to tell everybody about the grades. So these grades that show up in the grade center, these are just raw numbers. Just please make sure you don't try to determine your grade based on that, because I just take the numbers and then I put them in my Excel spreadsheet offline and I calculate each exam. Everything is going to get you know, their value according to the grading criteria that I have given you. Regarding grade in there, I think I want to see the grade because I want to know that people have done it. So yes, it shows me the grade in it. Let me see. It's checked for multiple attempts. I don't know why it's not showing up. Somebody said they couldn't do it.
Yes.
No. All right. Let me just change the date. There is a due date in 2 places. And maybe that's why I was interfering. But it is set up for multiple attempts. Okay. So I'm doing the same thing for the second practice quiz for this week. Making sure that you do have access to multiple attempts. Okay. So week 2. Test options. Edit the test options. No, it is allowed multiple. It's multiple.
Two connect
Limited.
assignments. It's a 3 connect assignments.
Are you saying something to me?
Yes, Professor, are you only doing, when you go to the connect, are you only doing the lecture exam 1 assignment, or you are doing both the lab? I see there is this, because my start date for the lab simulations, it starts from January 30th, 2021 to May 1st. And then I see one on February 18th to May 31st. So which one are you doing? Are you doing both the two?
The beginning date doesn't matter. Basically, all the lab simulations, they're not working right now anyway. But the lab simulation have no deadlines and they will be available throughout the semester.
Okay, so are you doing that one too, or you are doing only the lecture assignment?
She said only the lecture. Lab is not required. That's what she said.
Okay.
And also, please, I make sure that you print out the assignments sheet, the schedule that I have given you. It answers most of questions that you guys are asking me. If it is not listed in that syllabus schedule for lab and lecture, that means you don't have to worry about it. Okay? Whatever is mandatory is listed there. Any other questions? I hope I answered your question adequately. Did I?
Yes, Professor, that's okay. I do understand.
Okay. So both, all the practice quizzes for this and last week, they do have multiple attempts allowed. I don't know why it was not, but you should have multiple attempts access to it. Okay. Any other questions? Is everybody good?
Yes, for now.
Okay, so any other question, please do not wait until next week. When you're working and you have any issue, you can email me and I will get back to you in business hours, okay? If you call me or email me at 12 a.m. or like 1 a.m., of course I'm not going to answer. I'll answer the following day. But you will hear from me within a day.
Okay.
All right.
Okay,
so if
Professor.
everything is okay now, I'm going to stop.